Welcome to Fictional Scientist. After this video, reality will never feel the same again. Right now, at this exact moment, as you inhale and exhale without thinking about it, trillions of living organisms inside your body are making decisions for you. They are regulating your metabolism, training your immune system, shaping your cravings, modulating your mood, keeping you alive in ways you will never consciously perceive. Without them, you wouldn't last minutes, you wouldn't last seconds. So ask yourself the uncomfortable question, who is really living in this body? This is not a metaphor. This is biology. You were taught to think of yourself as one person, one mind, housed in one body, a single unified self moving through the world with clear boundaries. But that picture is a simplification, useful, comforting, and deeply misleading. It's a story your brain tells you to impose order on overwhelming complexity, because the truth is harder to live with. In truth, you are not a single being. You are a colony, a federation, a living, breathing ecosystem made up of trillions of cells, bacteria, viruses, fungi, and parasites, most of which are not even human. They outnumber your human cells. They outnumber your human genes, and they have been with you longer than your conscious mind ever could. Your body is not a kingdom ruled by a solitary monarch issuing commands from the brain. It is a parliament of microscopic life forms, each with its own agenda, its own survival strategy, its own chemical language. They negotiate, they cooperate, they compete, they influence outcomes. And the you that you think of as yourself, you are not the ruler of this system. You are its spokesperson, the press secretary of a biological government you did not elect and do not fully control. Tonight, we are going to dismantle the myth of the individual, not as a philosophical exercise, but as a scientific one. We are going to follow the evidence into territory most people instinctively resist, the realization that the boundaries of the self are far blurrier than you were taught, that your thoughts, your cravings, your moods, and even your decisions may not originate where you think they do. This is not speculation. This is not metaphorical storytelling. This is microbiology. This is neuroscience. This is the cutting edge of what it means to be human. And it begins with a single, undeniable fact. You are outnumbered. Let's start with something that sounds simple. A head count. A basic question that feels like it should have a clean, definitive answer. How many cells are in your body? For a long time, scientists believed they already knew. The estimate hovered around 10 trillion, a number repeated in textbooks, lectures, and documentaries for decades. It felt precise enough, comfortably large, respectable, but it was wrong. As measurement techniques improved and biological models became more accurate, that estimate quietly collapsed. Recent research now suggests the true number is closer to 37 trillion human cells. 37 trillion. That's 37 followed by 12 zeros. A number so vast that it stops functioning as information and becomes abstraction. You can't picture it. You can't intuitively compare it to anything familiar. The human mind simply wasn't built to hold numbers at that scale. But here's the problem. That number, as staggering as it is, still tells only half the story. For every human cell in your body, there is at least one non-human cell. Bacteria, viruses, fungi, archaea. These organisms are not confined to one hidden corner of your biology. They exist everywhere, on your skin, coating your teeth, lining your airways, and most densely, by far, in your gut. When scientists count the total number of microbial cells living in and on the human body, the result is striking. Their numbers are roughly equal to human cells. Some studies place the ratio at 1 to 1. Others suggest it tilts even further, closer to 1.3 to 1, in favor of the microbes. The exact ratio may vary slightly depending on the method, but the implication does not change. The conclusion is unavoidable. You are not a majority in your own body. You are a walking ecosystem, a mobile biome, a planet populated by trillions of organisms, most of which you will never see, never meet, and never consciously acknowledge. Yet they are undeniably present right now, living, feeding, reproducing, and dying on you, in you, and as you. And the strangest part, you need them. Without them, you would not survive. Let's make this concrete. The collective mass of all the microbes in your body, your microbiome, weighs between 1 and 3 kilograms. That's roughly the weight of your brain. Think about that. You are carrying around an entire kilogram of living, non-human organisms inside you at all times. A silent passenger, a stowaway civilization. But unlike a stowaway, they are not hiding. They are working. They are integrated into every system of your body. They are part of the infrastructure. Your skin, for instance, is home to roughly 1 trillion bacteria. They live in the follicles of your hair, in the creases of your knuckles, in the oils of your face. Some of them produce antimicrobial compounds that kill invaders. Others break down the oils your skin secretes, preventing fungal infections. They are your first line of defense, a living shield. Your mouth contains over 700 species of bacteria. They coat your teeth, your tongue, your gums. Some of them cause cavities, others prevent them. They form biofilms, complex multi-species communities that communicate with each other through chemical signals. They are not random, they are organized, they have structure, they have roles. Your gut is even more astonishing. The human intestine is home to an estimated 100 trillion microbial cells. This is the densest microbial ecosystem on Earth. Pound for pound, your colon contains more bacterial biomass than soil, than seawater, than any environment we've ever studied. And these bacteria are not passive. They are not just sitting there doing nothing. They are active participants in your biology. They digest your food, they synthesize vitamins, they regulate your immune system, they communicate with your brain. They are, in a very real sense, part of you. But here's the question that should keep you awake. If they are part of you, then where do they end and you begin? Now let's talk about genes. The human genome contains roughly 20,000 protein-coding genes. This is fewer than a tomato, fewer than a water flea. For a long time, this was embarrassing to scientists. We assumed that complexity required a large genome. But humans, despite all our sophistication, have a surprisingly small genetic toolkit. But here's the twist. Your microbiome contains roughly 5 million unique genes. That's 250 times more genetic information than your human cells carry. So when we talk about your biology, we need to ask, whose genes are we talking about? If you were to sequence all the DNA in your body right now, every strand, every chromosome, every plasmid, only 1% of it would be human. The other 99% would belong to bacteria, viruses, fungi, and other microbes. You are not a human being hosting some bacteria. You are a bacterial ecosystem hosting some human cells. This is not an exaggeration. This is the data. And it forces us to reconsider a very simple question. What does it mean to be you? You were not born alone. From the very first moment of your arrival into the world, you were already part of a living exchange. As you emerged from your mother's womb, your body was not a blank slate, not sterile, not empty. It was immediately claimed, coded, and welcomed by life. Passing through the birth canal, you were covered in a dense layer of bacteria, primarily lactobacillus species, the same microbes commonly found in yogurt and other fermented foods. These organisms were not accidents, they were not contaminants, they were a deliberate transfer, a gift, a biological dowry passed from one generation to the next. Within minutes of birth, your skin, your mouth, and your gut were seeded with microbial life. Your mother's microbiome became your microbiome, forming the foundation of the ecosystem that would grow with you for the rest of your life. This transfer is so critical that when it doesn't happen, the consequences are measurable. Babies born via cesarean section who bypass the birth canal entirely develop distinctly different microbial communities, and those differences matter. Statistically, C-section babies have higher rates of asthma, allergies, and autoimmune disorders later in life. Not because the surgery itself is harmful, but because something essential was skipped. the first exposure, the first inoculation. They missed the inheritance. Your first meal deepened this transfer. It was also your first deliberate dose of bacteria. Human breast milk is not sterile. It never was. It contains hundreds of bacterial species, roughly 700 identified so far, along with complex sugars called oligosaccharides that human infants cannot digest at all. These sugars are not meant for you. They pass through your system untouched. Their purpose is different. They exist to feed the bacteria that are taking root in your gut. Breast milk is not just nourishment, it is a prebiotic system engineered by evolution to cultivate the microbial community that will help shape your immune system, your metabolism, and even your brain. Your mother was not just feeding you, she was feeding your ecosystem. And this process does not end in infancy. It never truly stops. Throughout your life, with every meal you eat, every surface you touch, every breath you take, you are exchanging microbes with the world around you. Bacteria enter, bacteria leave, some stay, some disappear. You are not a closed container. You are not sealed off from your environment. You are porous, permeable, open. You are constantly becoming. But this is not a one-way relationship. This is not parasitism. This is symbiosis. The history of human evolution is not the history of a single species rising out of the mud. It is the history of a partnership, a coevolution. Humans did not evolve alone. We evolved together with our microbes. Consider the appendix. For a long time, we thought it was vestigial, a useless leftover from a herbivorous ancestor. But we were wrong. The appendix is a microbial safe house. When your gut is flushed by diarrhea or infection, the appendix serves as a reservoir, a bunker where beneficial bacteria can hide and then repopulate your intestine once the danger has passed. Your body is designed to preserve them, because without them, you are vulnerable. Or consider your immune system. For decades, scientists were baffled by autoimmune disorders. Why would the body attack itself? The answer, it turns out, involves the microbiome. Your immune system is not just trained to recognize foreign invaders, it is trained by your microbiome. The bacteria in your gut teach your immune cells the difference between friend and foe. If you grow up in an overly sterile environment, if you are never exposed to a diverse range of microbes, your immune system becomes confused. It starts attacking harmless substances, pollen, peanuts, gluten, your own tissues. The hygiene hypothesis is not about being dirty, it's about being microbially educated. And this education begins in infancy and continues throughout your life. Your microbiome is constantly sending signals to your immune cells, calibrating their responses, telling them when to attack and when to stand down. Your microbiome is not just living in you, it is teaching you. And then there are the viruses. For most people, viruses exist in a single category: enemies, invaders, pathogens to be feared, avoided, eliminated. They are associated with illness, outbreaks, and loss of control. But this view captures only a small fraction of the truth. The deeper story is far more unsettling and far more intimate. Roughly 8% of the human genome is viral DNA. Not metaphorically, literally. Embedded in your chromosomes are the genetic remnants of ancient viral infections, retroviruses that once invaded the cells of your ancestors millions of years ago. These were not fleeting encounters. These viruses inserted their genetic material directly into the DNA of reproductive cells, ensuring that their sequences would be passed down generation after generation. Over evolutionary time, these viral fragments lost their ability to form infectious particles. They were neutralized, tamed, but they were not discarded. Instead, many of these viral sequences were domesticated, repurposed, integrated into the machinery of human biology. What began as an invasion slowly became infrastructure. Viral genes were co-opted, modified, and put to work in service of the host. Some of these genes are now indispensable. One of the most striking examples is syncytin, a gene derived from an ancient retrovirus. Syncytin plays a critical role in the formation of the placenta. enabling fetal cells to fuse with maternal tissue and create the barrier that allows pregnancy to proceed. Without this viral gene, the placenta cannot form properly. Without it, human pregnancy, as well as pregnancy in all placental mammals, would be impossible. We do not simply coexist with this virus, we depend on it. We quite literally owe our existence to a viral infection that occurred in a mammalian ancestor roughly 150 million years ago. But the viral story does not end in deep time. It is not confined to fossils written in DNA. Right now, in this moment, your body is home to trillions of active viruses. Most of them are bacteriophages, viruses that infect bacteria rather than human cells. In your gut alone, these phages outnumber bacteria by as much as 10 to 1. They are not passive passengers. They are active regulators. They infect bacterial cells, replicate inside them, destroy them, and in doing so, shape which bacterial populations survive and which collapse. Through this constant pressure, bacteriophages sculpt the structure of your microbiome. They prevent any single bacterial species from dominating. They maintain balance. They enforce turnover. They act as invisible predators in a microscopic ecosystem, keeping the system dynamic, adaptable, and alive. You are not just a human-bacteria hybrid. You are a human-bacteria-virus chimera, and we are only beginning to understand what this means. Your gut is often called the second brain, but this is misleading. It suggests the gut is secondary, subordinate, a backup system. In truth, your gut may be the first brain. The enteric nervous system, the network of neurons lining your intestines, contains roughly 500 million neurons. That's more than your spinal cord, more than the peripheral nervous system. It is the second largest concentration of neural tissue in your body, and it operates independently. You do not consciously control digestion. You do not tell your intestines to contract, your stomach to churn, your pancreas to release enzymes. These processes are autonomous, reflexive, managed by the gut itself. But here's where it gets strange. Your gut is in constant communication with your brain. The vagus nerve, a thick bundle of nerve fibers running from your brainstem to your abdomen, is the main highway of this communication. And the traffic is not one way. Your brain sends signals to your gut, yes, but your gut sends far more signals to your brain. Roughly 90% of the signals traveling along the vagus nerve are ascending from gut to brain, not the other way around. Your gut is not listening to your brain. Your brain is listening to your gut. And what is your gut saying? It turns out your gut bacteria are doing far more talking than you ever realized. What feels like an internal monologue, a private chemical conversation inside your brain, is in many cases being initiated somewhere else entirely. Deep in your digestive tract, microbial communities are actively generating signals and sending messages upward, shaping how you feel long before you become aware of it. Certain bacterial species are capable of producing neurotransmitters, the very same chemical messengers your brain uses to communicate between neurons. This is not a loose analogy. It is the same molecular language. Species such as lactobacillus and bifidobacteria manufacture gamma-aminobutyric acid, better known as GABA. GABA is the brain's primary inhibitory neurotransmitter. It dampens excessive neural firing. It lowers anxiety. It stabilizes mental noise. It is, chemically speaking, the signal that tells your nervous system to slow down. Other bacteria influence the production of serotonin, and here the numbers are impossible to ignore. Roughly 90% of the body's serotonin is produced in the gut, not in the brain. Serotonin regulates mood, appetite, sleep cycles, motivation, and social behavior. It plays a central role in how you experience pleasure, satisfaction, and emotional balance. It is one of the most important signaling molecules in your entire body, and the majority of it exists because gut microbes stimulate its production. Let that sink in. When you feel happy, when you feel calm, when you feel satiated, some of that feeling is not coming from you. It is coming from the microbes in your gut. But it goes further, much further. Certain bacteria can alter your behavior, and they do, deliberately. Take Toxoplasma gondii, a single-celled parasite that infects roughly one-third of the human population. Its primary host is the cat, but it can infect almost any warm-blooded animal, including humans. When it infects a rodent, it does something remarkable. It changes the rodent's behavior. Infected mice lose their fear of cats. In fact, they become attracted to the smell of cat urine. This is not a random side effect. This is adaptive manipulation. The parasite is increasing the likelihood that the mouse will be eaten by a cat, allowing the parasite to complete its life cycle in the cat's gut. And humans. Infected humans show subtle but measurable changes in personality. Men tend to become more introverted, suspicious, and rule-breaking. Women tend to become more outgoing, trusting, and conscientious. The mechanisms are not fully understood, but the data is clear. The parasite is altering human behavior. And Toxoplasma is not alone. Certain gut bacteria have been linked to depression, others to anxiety, others to autism spectrum disorders. Researchers have transferred the gut microbiome from depressed humans into germ-free mice. Mice raised in sterile conditions with no microbiome of their own. The mice developed depression-like behaviors. They became lethargic. They gave up more easily in stress tests. They showed all the hallmarks of despair. The bacteria carried the behavior. Now we must ask, how many of your own behaviors are actually yours? How many of your moods are generated by your neurons? And how many are whispered into your brain by bacterial messengers? How much of you is actually them? Last night you felt a craving. Maybe it was chocolate, maybe bread, maybe something salty, greasy, sweet. It appeared suddenly, almost reflexively. A pull, a desire, a quiet urgency that felt physical, undeniable. Hunger rising not just in your stomach, but in your attention, and you assumed it was you. But was it? Your gut bacteria survive on what you eat. Every species has preferences. Some thrive on fiber, others specialize in fat, others flourish when sugar is abundant. And here's the part most people never consider. They don't just passively consume these nutrients. They compete for them, they adapt to them, and they push for more of whatever keeps them alive. Bacteria that feed on sugar don't wait quietly for their next meal. They send signals. Chemical requests routed through your nervous system. Bacteria that thrive on fat do the same. They influence your brain by producing neurotransmitters, by activating reward circuits, by adjusting hunger and satiety hormones. They shape what feels appealing, what feels urgent, what feels satisfying. They're lobbying your brain. This isn't speculation. Studies consistently show that people with a highly diverse gut microbiome, those whose intestines host a wide range of bacterial species, tend to experience fewer cravings overall. Their appetites are more stable. Their hunger signals are less extreme. They are less prone to binge eating or obsessive food desires. In contrast, people with low microbial diversity, where only a handful of bacterial species dominate, often experience intense, narrow cravings that feel almost compulsive. Because when only a few species are in charge, their signals get louder, because the bacteria are screaming, and it goes deeper than preference alone. Researchers have identified specific bacterial metabolites, chemical byproducts released as bacteria digest food, that directly influence appetite regulation. When bacteria ferment dietary fiber, they produce short-chain fatty acids, or SCFAs, such as butyrate and propionate. These molecules don't stay in the gut, they enter the bloodstream, they travel to the brain, and when they arrive, they deliver a message: you've had enough. They signal satiety, they tell you you're full. But if your diet lacks fiber, your bacteria can't produce enough of these molecules. The signal never arrives, the feedback loop breaks, and so you keep eating. You feel hungry, not because your body needs more calories, but because your bacteria need more fiber. Your hunger is not always yours. Let's get specific. Chocolate. People often joke about being addicted to chocolate, but there's a biological basis for this. Chocolate contains compounds that certain gut bacteria love: polyphenols, flavonoids, and theobromine. When you eat chocolate, these bacteria feast, and in return, they produce metabolites that trigger dopamine release in your brain. You feel pleasure, you feel reward, you want more. But here's the question, are you eating chocolate because you enjoy it, or because your bacteria have trained you to enjoy it? There is evidence that people with a strong preference for chocolate have different gut bacteria than people who don't care for it. The bacteria shape the preference, they create the craving, and then they satisfy themselves when you give in. This is not a metaphor, this is manipulation. And it's not limited to chocolate. The same principle applies to alcohol. People with alcohol use disorder have distinct microbial signatures. Their gut bacteria produce metabolites that increase cravings, that amplify the rewarding effects of alcohol, that make it harder to quit. The bacteria are codependent, and they drag you down with them. Now let's talk about probiotics. The probiotic industry is worth billions of dollars, built on a promise that sounds almost irresistible. Consume these good bacteria and your health will improve. Your digestion will normalize, your mood will stabilize, your gut will heal itself. It's a clean, comforting narrative. One capsule, one drink, one solution. But beneath the marketing language and colorful packaging, the science tells a far more complicated story. First, most probiotic bacteria do not actually colonize your gut. They do not move in. They do not settle down. In most cases, they simply pass through. You ingest them, they struggle through the acidic environment of your stomach. If they survive at all, they briefly interact with the microbial communities already living inside you, and then they are gone. Hours later, sometimes days, they are expelled. They do not become part of your ecosystem. They leave no lasting footprint. They are visitors, tourists, not residents. Second, not all bacteria are beneficial to all people. There is no universally helpful strain. A bacterial species that improves digestion or reduces inflammation in one person may cause bloating, discomfort, or worse in another. The microbiome is deeply personal, shaped by genetics, early life exposure, diet, environment, and history. It is as individual as a fingerprint. What works for one host may disrupt balance in another. The same intervention can heal one ecosystem and destabilize the next. Third, some bacteria marketed as probiotics can be harmful in certain contexts. Lactobacillus, for example, is often portrayed as a model citizen of the gut. Safe, beneficial, friendly. And for many people it is. But in others, especially those with compromised immune systems or damaged intestinal barriers, lactobacillus can escape the gut and enter the bloodstream, where it becomes dangerous. In rare but real cases, it can cause serious infections. The line between symbiont and threat is not fixed. It depends entirely on context. The idea of a universal good bacteria is a myth. Context matters. Ecology matters. The microbiome is not a simple garden where you can plant a few seeds and expect predictable results. It is a jungle, a battlefield, a complex adaptive system by competition, scarcity, cooperation, and constant change. It resists control, it resists simplification, and the bacteria know this. They are not passive. They are constantly competing and cooperating. They're sending chemical signals, forming alliances, and turning against one another when conditions shift. They wage microscopic wars using toxins and antibiotics. They build protective fortresses out of biofilm. They exchange genes like weapons and tools. They adapt, they evolve, and they do all of this in real time inside your body. You are not managing your microbiome. Your microbiome is managing itself, and you are just along for the ride. Your immune system has a problem. A paradox. On one hand, it must be aggressive. It must recognize and destroy anything foreign. Bacteria, viruses, fungi, parasites, these are threats. Invaders, enemies. The immune system's job is to find them and kill them. But on the other hand, your body is full of foreign organisms, trillions of them, and most of them are not enemies. They are allies, partners, symbionts. So how does your immune system know the difference? The answer is it learns, but not from you. It learns from them. When you are born, your immune system is naive, uneducated. It has the capacity to recognize foreign molecules, antigens, but it doesn't yet know which ones are dangerous. It needs training, and the primary trainers are your gut bacteria. In the first few years of life, your microbiome teaches your immune system what is normal, what is safe, what belongs. This education happens in the gut-associated lymphoid tissue, GALT, a network of immune cells embedded in the lining of your intestines. These cells sample the bacteria in your gut. They interact with them, they learn from them, and this learning is permanent. The immune system forms a memory, a list of approved residents, a treaty. As long as the bacteria stay in the gut, as long as they don't cross the intestinal barrier into the bloodstream, they are tolerated, protected, allowed to live. But if the treaty is broken, if the barrier is breached, the immune system attacks. The intestinal barrier is only one cell thick. A single, fragile layer of epithelial cells stands between the trillions of bacteria living in your gut and the sterile environment of your bloodstream. There is no armor here, no buffer zone, just a microscopic wall doing one of the most important jobs in your body. This barrier is held together by structures known as tight junctions, specialized protein complexes that seal the spaces between neighboring cells, preventing unwanted passage while still allowing nutrients to be absorbed. But these junctions are not fixed in place. They are not rigid bricks in a wall. They are dynamic, responsive, constantly opening and closing in reaction to chemical signals. And one of the most powerful influences on their behavior comes from something deeply familiar: your diet. A diet high in processed foods, refined sugar, and unhealthy fats sends the wrong signals. Over time, these signals weaken the tight junctions. The seals loosen. Microscopic gaps begin to form between cells, and through these gaps, bacteria and bacterial toxins slip through. Among the most dangerous of these toxins are lipopolysaccharides, or LPS, molecules that form part of the outer membrane of certain bacteria. When LPS crosses the intestinal barrier and enters the bloodstream, it does not go unnoticed. This state is commonly referred to as leaky gut syndrome, and when it occurs, the immune system reacts immediately and aggressively. LPS is one of the most potent immune activators known to biology. Its presence in the blood triggers alarm systems that evolve to detect life-threatening infection. White blood cells are rapidly mobilized. Inflammatory cytokines are released in large quantities. The immune system shifts into combat mode. The body prepares for war. But there is a problem. The enemy is no longer localized. The breach is not confined to a single point. The bacteria are everywhere. The wall has failed, and the immune system cannot determine where the invasion began or where it will end. So it responds indiscriminately. It attacks broadly, relentlessly. It becomes hyperactive, paranoid, aggressive. What begins as a defensive reaction turns into a permanent state. Chronic inflammation sets in, and chronic inflammation lies at the root of nearly every major disease of modern life: heart disease, diabetes, Alzheimer's, autoimmune disorders, cancer, depression, different conditions, different organs, the same underlying fire. The common thread is a breakdown in the immune microbiome treaty, a loss of trust, a collapse of the negotiation. Your body is at war with itself, and the bacteria are caught in the crossfire. But sometimes the immune system doesn't just attack bacteria, it attacks you. Autoimmune diseases, conditions where the immune system targets the body's own tissues, are on the rise. Type 1 diabetes, rheumatoid arthritis, multiple sclerosis, lupus, Crohn's disease, celiac disease. The list is long and growing, and in almost every case, the microbiome is involved. Take type 1 diabetes. This is a condition where the immune system destroys the insulin-producing cells in the pancreas. For a long time, we thought it was purely genetic. But identical twins who share 100% of their DNA only have a 50% concordance rate for type 1 diabetes. If one twin develops it, The other twin has only a 50% chance of developing it. Genes are not the whole story. Environment matters. And the most important environmental factor is the microbiome. Studies have shown that children who develop type 1 diabetes have distinct microbial signatures months or even years before the disease manifests. Their gut bacteria are less diverse, certain beneficial species are missing, others are overrepresented. The theory is that these microbial imbalances trigger a cascading immune dysfunction. The immune system, poorly educated by a dysfunctional microbiome, begins attacking not just bacteria, but anything that resembles bacterial proteins, including the body's own cells. This is molecular mimicry. The immune system is fooled. It sees a resemblance between a bacterial protein and a human protein, and it cannot tell the difference, so it attacks both. Your immune system is not betraying you. It is confused, misled by bad intel from a corrupted microbiome, and the cure is not more drugs. It is restoration, re-education, a rebuilding of the microbial community, a return to the treaty. Your microbiome is not a random collection of bacteria assembled by chance. It is a record, a living archive, a history written not in stone or ink, but in microbial DNA. Every strain, every species, every genetic fragment carries information about where you've been, how you've lived, and what you've been exposed to over the course of your life. Every place you've lived leaves a trace. Every person you've touched, every surface you've shared, every meal you've eaten contributes to the shifting composition of your microbiome. This record is so detailed that researchers can analyze a person's gut bacteria and determine with surprising accuracy where they were born, what kind of diet they follow, whether they live in an urban environment or a rural one, and even whether they share their home with animals. The microbes remember what you forget. They keep track of your environment long after you've moved on. Your microbiome is a microbial fingerprint. Unique, personal, revealing. But it is not only a record of your own life, it is also a record of lives that came before you. Certain bacterial strains are passed down across generations, quietly, persistently, from mother to child, from grandmother to grandchild. These are heirloom microbes, inherited not through chromosomes, but through contact, care, and shared environments. They carry the ecological memory of your lineage. Encoded within them is information about the landscapes your ancestors inhabited, the foods that sustained them, and the diseases they endured and survived. In indigenous populations, communities that have remained in the same regions for thousands of years, this microbial inheritance is especially visible. Their microbiomes contain deep, ancient signatures. Bacterial strains that are rare or entirely absent in industrialized societies remain abundant and stable in these groups. These microbes are finely adapted to traditional diets, local water sources, and specific environmental conditions. They represent a continuity that stretches back through countless generations. But in industrialized populations, this continuity is breaking down. Microbial diversity is collapsing. Each generation carries fewer bacterial species than the one before. Strains that accompanied humanity for millennia are quietly disappearing, and unlike lost memories or forgotten skills, once these microbes are gone, they cannot simply be recovered. This is microbial extinction, and it is happening inside you. The single greatest cause of this extinction is antibiotics. Antibiotics are miracle drugs. They have saved hundreds of millions of lives, but they are not precision weapons. They are carpet bombs. When you take an antibiotic, it does not selectively target the pathogenic bacteria causing your infection. It kills everything: beneficial bacteria, neutral bacteria, bacteria that have been with your family for generations. All of them are wiped out. And while some of these bacteria recover after the antibiotic course is finished, others do not. Some bacterial species once lost do not return. The ecosystem is permanently altered. Studies have shown that a single course of antibiotics can reduce microbial diversity for months. Multiple courses, common in childhood, can cause long-term, perhaps permanent changes to the microbiome. And these changes have consequences. Children who receive multiple courses of antibiotics in early life have higher rates of obesity, asthma, allergies, and inflammatory bowel disease. The microbiome is destabilized. The ecosystem is thrown into chaos. Opportunistic bacteria, species that thrive in disturbed environments, take over. Diversity collapses, and once diversity is lost, it is incredibly difficult to restore. This is not an argument against antibiotics. When they are needed, they are essential, but they are overprescribed, overused, treated as a first-line defense for conditions that would resolve on their own. And every time we use them unnecessarily, we are eroding the microbial foundation of our health. We are burning the library to stay warm. Some bacteria have already been lost, not hypothetically, not in theory, actually lost, erased from large portions of the human population before we fully understood what they were doing for us. Their disappearance went mostly unnoticed, framed as progress, celebrated as a medical victory. One of the most striking examples is Helicobacter pylori, a bacterium that lives in the human stomach. For most of the 20th century, H. pylori was classified as an enemy. It was linked to gastric ulcers. It was associated with an increased risk of stomach cancer. Once its role in these conditions was identified, the medical response was clear and decisive: eliminate it. And we did. In industrialized countries, H. pylori infection rates collapsed. In the United States, fewer than 6% of children now carry the bacterium, down from more than 50% just a few decades ago. Antibiotics worked. Public health campaigns succeeded. By conventional standards, this was a triumph. Mission accomplished, right? Not quite. As it turns out, H. pylori is not merely a pathogen. It is an ancient human symbiont, one that has accompanied us for at least 100,000 years. Over that time, it became woven into our biology, and while it can cause disease in some individuals, in others it plays a protective role. People who carry H. pylori have lower rates of asthma and allergies. They are less likely to develop certain forms of esophageal cancer. The bacterium helps regulate stomach acid production. It shapes immune responses during early development. It participates in the normal functioning of the human microbiome. By eradicating it, we didn't eliminate disease, we rearranged it. We reduced stomach ulcers, yes, but we saw increases in asthma and allergic disorders. We solved one problem and quietly created two more. A trade was made, whether we intended it or not. This is the danger of viewing bacteria as purely pathogenic, of treating the microbiome as an enemy to be conquered rather than an ecosystem to be understood, of applying simple solutions to complex systems, of using a hammer where precision was required. We are losing bacterial strains faster than we can study them. Entire lineages are disappearing before their functions are fully mapped. And we don't know what we are losing. We don't know which microbes are essential, which are redundant, and which are keystone species holding the entire system together. By the time we figure it out, it may be too late. Let's return to serotonin, because this is where the story gets personal. Serotonin is often called the happiness molecule. This is an oversimplification, but it's not entirely wrong. Serotonin regulates mood, social behavior, sleep, appetite, and memory. Low serotonin levels are associated with depression, anxiety, and obsessive-compulsive disorder. Most antidepressant medications work by increasing serotonin availability in the brain. But here's the strange part. 90% of your body's serotonin is produced in your gut, not your brain, your gut. And it's not even produced by your human cells. It's produced by enterochromaffin cells in the lining of your intestines, cells that are heavily influenced by gut bacteria. Certain bacterial species stimulate serotonin production, others inhibit it. The composition of your microbiome directly affects how much serotonin your gut produces. And while most of this gut-derived serotonin does not cross the blood-brain barrier, it doesn't need to. It sends signals through the vagus nerve. It communicates with the brain indirectly. Your gut bacteria are modulating your mood, remotely, chemically, constantly. And when the microbiome is disrupted by antibiotics, by stress, by a poor diet, serotonin production drops. The gut-brain axis goes offline. Mood destabilizes. Depression is not just a brain disorder, it is a whole body disorder, an ecosystem disorder. Now let's talk about anxiety. Anxiety disorders are the most common mental health conditions in the world. Hundreds of millions of people live with chronic, debilitating anxiety, and for most of them, the cause is unknown. Genetics plays a role. Trauma plays a role. But these do not explain the global rise in anxiety over the past few decades. Enter the microbiome. Researchers have identified specific bacterial species that are associated with anxiety. One of the most studied is Lactobacillus rhamnosus. When this bacterium is given to mice, it reduces anxiety-like behavior. Mice spend more time in open spaces, they explore more, they show fewer stress responses, and the effect is mediated by GABA receptors in the brain, the same receptors targeted by anti-anxiety medications like Xanax and Valium. But here's the twist. If the vagus nerve is severed, the effect disappears. The bacteria can no longer communicate with the brain. The anxiety returns. This proves that the bacteria are not acting on the brain directly. They are sending signals through the vagus nerve. They are talking to the brain, and the brain is listening. Other studies have shown that people with anxiety disorders have lower microbial diversity. Their guts are dominated by inflammatory bacterial species that produce metabolites that activate the stress response. These people are not imagining their anxiety. They are not weak. They are not broken. They are living in a hostile microbial environment. Their gut bacteria are generating stress signals, and their brains are responding accordingly. The anxiety is real, but the source is not where they think it is. And then there is depression. Major depressive disorder affects over 300 million people worldwide. It is one of the leading causes of disability, and despite decades of research, we still don't fully understand it. Antidepressants work for some people, but not for others. Therapy helps, but not always. What if we've been looking in the wrong place? In 2016, researchers conducted a landmark study. They took fecal samples from people with major depression and transplanted them into germ-free mice with no microbiome of their own. The mice developed depression-like behaviors. They became lethargic. They stopped grooming. They gave up more quickly in stress tests. They exhibited anhedonia, the inability to experience pleasure. The bacteria carried the depression. When the researchers transplanted fecal samples from healthy controls into germ-free mice, the mice remained healthy, active, curious. The difference was the microbiome. Follow-up studies identified specific bacterial metabolites that are elevated in depressed individuals. One of them is a molecule called indole. Indole is produced when certain bacteria break down the amino acid tryptophan, the same molecule your brain uses to make serotonin. When bacteria consume too much tryptophan, less is available for serotonin synthesis. Mood drops. Another metabolite is lipopolysaccharide, LPS, the same immune-activating molecule we discussed earlier. LPS triggers inflammation, and chronic inflammation is one of the strongest predictors of depression. People with depression have higher levels of inflammatory markers in their blood. Their immune systems are overactive, and the source of the inflammation is often the gut. A leaky gut, a dysfunctional microbiome, bacteria in the wrong place, doing the wrong things. Depression in many cases is an immune disorder, an inflammatory disorder, a microbial disorder, and treating it requires more than just medication. It requires ecological restoration. Let's zoom in even further. Let's go inside your cells. Every human cell except red blood cells contains mitochondria. These are the powerhouses of the cell. They convert nutrients into ATP, the energy currency of life. Without mitochondria, complex life would not exist. You would not exist. But here's the secret. Mitochondria are not human. They are not even eukaryotic. They are bacteria. About 1.5 billion years ago, a larger cell engulfed a smaller bacterium. Instead of digesting it, the larger cell kept it. And the bacterium, instead of dying, began to cooperate. It provided energy. The host cell provided shelter. Over time, the bacterium became permanently integrated. It lost its independence. It became an organelle. But it never forgot its origins. Mitochondria still have their own DNA, circular bacterial DNA. They reproduce independently of the cell. They have double membranes, a relic of the original engulfing event. They are prisoners or partners or both. This event, endosymbiosis, was the most important evolutionary breakthrough in the history of life. It allowed cells to become larger, more complex, more energy efficient. It made animals possible. Plants, fungi, all complex life on Earth descends from that single act of cooperation. You are not a pure human, you are a chimera, a fusion of multiple ancient life forms. And the same is true of plants. Chloroplasts, the organelles that perform photosynthesis, are also bacteria, cyanobacteria, to be specific. They too were engulfed. They too became permanent residents. Every breath you take, every movement you make is powered by ancient bacteria living inside your cells. They are not guests, they are not parasites, they are foundational. And this raises a question. If your cells are made of bacteria, where does you begin? And then there are the viruses in your DNA. We've already noted that roughly 8% of the human genome is viral in origin. But that number alone doesn't capture what's really happening. These sequences, known as endogenous retroviruses, or ERVs, are not just genetic debris left behind by ancient infections. They are not useless fragments drifting through your chromosomes. Some of them are active, functional, indispensable. One of the most important of these viral genes is syncytin. Syncytin is responsible for the formation of the syncytiotrophoblast, the specialized layer of cells that becomes the placenta. This structure allows nutrients, oxygen, and waste to pass between mother and fetus while keeping their blood supplies separate. Without syncytin, the placenta cannot form. Without a placenta, pregnancy cannot continue. And without pregnancy, mammals, including humans, would not exist. And syncytin is viral. Originally, it was an envelope protein used by retroviruses to fuse with host cells during infection. At some point deep in mammalian evolutionary history, this viral gene was captured by the host genome. Instead of being eliminated, it was preserved, modified, repurposed. What once served viral invasion became the biological foundation of reproduction itself. We owe our existence to a virus. And syncytin is not an isolated case. Other viral genes embedded in your DNA play roles in regulating immune responses, controlling when and where genes are switched on, and even defending against future viral infections by interfering with similar invaders. The viral sequences in your genome are not scars from past battles. They are tools, a molecular toolkit, a library of evolutionary solutions borrowed from ancient enemies and transformed into assets. You are not just a human-bacteria hybrid. You are a human-bacteria-virus mosaic, and this mosaic is not finished. New viruses are still integrating into the human genome. Evolution has not stopped. It is still happening quietly inside you. You are still becoming. There is a term for this, a concept that scientists are only beginning to take seriously. It's called the holobiont. A holobiont is an organism plus its microbiome treated as a single evolutionary unit. The host and its symbionts are not separate entities. They are a collective, a superorganism. They evolve together, they adapt together, they survive or perish together. You are not a human, you are a holobiont, a walking ecosystem, a federation of trillions of organisms, most of which are not human, all of which are you. And this has profound implications for how we think about health, disease, identity, and evolution. When a doctor treats you, they are not treating a single organism, they are treating an ecosystem. When you eat, you are not just feeding yourself, you are feeding a community. When you take a medication, you are not just affecting your human cells, you are affecting trillions of microbial cells, each with their own metabolism, their own vulnerabilities, their own responses. We have been treating the human body as a machine, a car that breaks down and needs to be repaired. But the body is not a machine. It is a garden. And gardens require a different kind of care. You cannot fix a garden with a hammer. You have to understand the soil, the seasons, the relationships between plants, the insects, the microbes, the water, the light. You have to understand the ecology. So let's finally ask the question we've been circling for the past 2.5 hours. Who are you? You were taught a simple story. You are a single being, a unified self, a body directed by a brain that generates a mind that experiences consciousness. You are the captain of your ship, the author of your story, the origin of your choices, your desires, your actions. Agency flows outward from you cleanly and decisively, but every piece of evidence we've examined undermines that story, or at the very least reveals how incomplete it is. You are not a single being. You are a collective, a parliament, a federation made up of trillions of organisms, human and non-human alike. Each has its own needs, its own survival strategies, its own influence. They vote chemically, they negotiate biologically, and together they shape what you experience as you. Your cravings are not entirely yours. Your moods are not entirely yours. Your immune responses are not entirely yours. Even your thoughts, those most intimate, most private experiences you assume belong exclusively to you, are being shaped continuously by microbial metabolites circulating in your blood, by hormones produced in your gut, by signals traveling along the vagus nerve from bacteria to brain. The line between internal and external dissolves the closer you look. So where is the boundary? Where does you end and them begin? The answer is uncomfortable but unavoidable. There is no boundary. At least not a clean one, not a stable one, not the kind you were promised. You are not a castle surrounded by walls, defending a fixed interior. You are a city with open borders. Organisms are constantly moving in and out. Genes are being exchanged. Signals are being sent, received, interpreted, and modified. Nothing is static. Nothing is isolated. You are not a noun. You are a verb, a process, a continuous negotiation between human and microbial cells. And this is not a metaphor. This is biology. Now let's take this to its logical conclusion. If your bacteria are influencing your behavior, your food choices, your mood, your social interactions, then how much free will do you actually have? This is not a comfortable question. We want to believe that we are in control, that our decisions are ours, that we are the authors of our own lives, but the evidence suggests otherwise, at least partially. Consider cravings again. You feel a desire for chocolate. You think it's your desire, but it's also the desire of the bacteria that thrive on chocolate. You feel a craving for alcohol, but it's also the craving of bacteria that produce metabolites that amplify the rewarding effects of alcohol. Your decision to eat chocolate or drink alcohol is influenced by forces you are not aware of, forces that are not you in the traditional sense. Does this mean you have no free will? No, but it means free will is more complicated than we thought. It is not a binary. It is not all or nothing. It is a spectrum, a negotiation, a collaboration. You are not a dictator ruling over your body. You are a diplomat negotiating with a parliament of microbial representatives, and sometimes the parliament wins. But here's the twist. This is not a tragedy. This is liberation. For centuries, we have framed human behavior as a moral failure. We have blamed individuals for their addictions, their moods, their lack of motivation, their inability to change. We told them to try harder, to be stronger, to exert more discipline, to take control, as if control were simply a matter of character, as if biology played no role at all. But if behavior is shaped in part by microbes, if cravings are intensified by bacterial signals, if depression is linked to chronic gut inflammation, if anxiety is modulated by signals traveling along the vagus nerve, then that entire framework collapses. The blame was never accurate. It was just convenient. People are not weak. They are not broken. They are living inside hostile or imbalanced microbial environments. And when the environment is hostile, no amount of willpower can override it for long. The problem is not moral failure. The problem is ecology. This is what compassionate science looks like. It begins by saying, you are not alone in your struggles. You are not solely responsible for your pain. Your symptoms are not personal flaws. They are signals emerging from a complex biological system. And systems, unlike character defects, can be understood. They can be adjusted. They can be repaired. This is not an excuse. It is an explanation. And explanations are powerful because explanations point towards solutions. If depression is partly microbial, then treatment doesn't have to stop at neurotransmitters alone. We can intervene through probiotics, prebiotics, dietary changes, and even fecal microbiota transplants. If anxiety is linked to dysregulation along the vagus nerve, then practices like slow breathing, meditation, and cold exposure become biological interventions, not vague self-help advice. If cravings are being driven by specific bacterial populations, then the strategy shifts. Starve the bacteria that fuel those cravings and nourish the ones that promote balance and satiety. We can change the parliament, we can elect new representatives, we can restore balance. And this reframing is deeply empowering because it means you are not trapped by who you are today. You are not doomed to repeat the same patterns forever. You are not defined by your current state or your past failures. Biology is not destiny when biology is dynamic. You are a garden, and gardens can be tended. So how do you tend the garden? The first step is obvious: diet. Your gut bacteria eat what you eat. If you eat a diet rich in fiber, fruits, vegetables, whole grains, legumes, you feed the bacteria that produce beneficial metabolites, short-chain fatty acids, vitamins, anti-inflammatory compounds. If you eat a diet rich in processed foods, sugar, and unhealthy fats, you feed the bacteria that produce inflammatory compounds. LPS, trimethylamine N-oxide, TMAO, indole. You are feeding a microbial army. The question is, which army do you want to win? Studies have shown that dietary changes can shift the microbiome within days. Within 24 hours of switching from a plant-based diet to a meat-based diet, the composition of gut bacteria begins to change. Inflammatory species increase, fiber-fermenting species decline. But the reverse is also true. Within days of increasing fiber intake, beneficial bacteria rebound. Diversity increases, inflammation decreases. Your diet is not just food for you, it is information for your ecosystem. It is a signal, a command, a vote, and every meal is a new vote. But diet is not only about what you eat, it is also about how your food is prepared. Long before supplements, before capsules, before nutrition labels, humans relied on fermentation. Foods like yogurt, kefir, sauerkraut, kimchi, miso, and tempeh are not modern health trends. They are some of the oldest foods in human history. For thousands of years, across cultures and continents, people fermented vegetables, dairy, grains, and legumes, not merely to preserve them, but to fundamentally transform them. Fermentation is a microbial process. It is bacteria and yeasts doing what they do best: breaking down sugars, producing acids, generating gases, creating new flavors, textures, and chemical landscapes. In the process, these microbes make food more digestible, more nutritious, and more biologically active. They also generate probiotics, living bacteria capable of surviving digestion and interacting with the gut ecosystem. But here is the critical distinction most discussions miss. Fermented foods do not simply contain probiotics. They contain entire ecosystems. Alongside live bacteria, they deliver prebiotics that feed microbes, postbiotics that influence immune and metabolic signaling, enzymes that aid digestion, vitamins synthesized during fermentation, and a dense web of microbial interactions. They are not defined by a single strain or species. They are communities, consortia, living systems. When you eat fermented foods, you are not just ingesting bacteria, you are ingesting a microbial culture in the fullest sense of the word. A living tradition passed down through generations. an ecological memory encoded in taste, smell, and biology. Research increasingly supports what traditional diets discovered long ago. People who regularly consume fermented foods tend to have greater microbial diversity, lower levels of systemic inflammation, and stronger immune responses. Fermented foods don't simply add microbes to the gut, they reorganize the ecosystem. They change the balance of power. They create conditions where beneficial species can thrive. And this matters because diversity is stability. A diverse microbiome is resilient. It can absorb shocks. It can recover after antibiotics, psychological stress, illness, or periods of poor diet. It is harder to destabilize and easier to repair. A diverse microbiome is a healthy microbiome. And then there is the most radical intervention of all, fecal microbiota transplantation, FMT. This is exactly what it sounds like. Doctors take stool from a healthy donor, process it, and transplant it into the gut of a patient with a disrupted microbiome. It sounds absurd, disgusting even, but it works astonishingly well. FMT is now the standard treatment for recurrent Clostridioides difficile infections, a severe, life-threatening bacterial infection that causes chronic diarrhea. Antibiotics are often ineffective, but FMT has a cure rate of over 90%. One treatment, one transplant, and the infection is gone. Because the transplanted microbiome outcompetes the pathogen, it restores the ecosystem, it rebuilds the wall. But researchers are now exploring FMT for other conditions: inflammatory bowel disease, obesity, diabetes, autism, depression. The results are mixed. Some patients improve dramatically, others see no change. The science is still young, but the principle is clear. The microbiome is not fixed, it can be replaced, reset, restored. You are not stuck with the bacteria you have. You can quite literally become someone else, microbially speaking. And this raises profound questions about identity, about personhood, about what it means to be you. If you receive a microbiome transplant and your mood changes, your cravings change, your behaviors change, have you become a different person, or have you simply restored your true self? There is no easy answer. But the question itself reveals the depth of the microbial influence. It reveals just how much of you is actually them. And now, as we come to the end of this journey, let's return to the question we started with. Who is really living in this body? The answer is everyone and no one. You are not a single being. You are a federation, a living ecosystem, a collective of trillions of organisms, most of which are not human, all of which are you. Your body is not a temple, it is a city, a bustling metropolis inhabited by countless species, each with their own role, their own needs, their own voice. And the you that you call yourself, the conscious, thinking, feeling self, is not the mayor of this city. You are the spokesperson, the ambassador, the face of the federation. You do not rule, you represent. And this is not a diminishment. This is an expansion. Because if you are not a single being, if you are a collective, then you are never truly alone. You are always in relationship, always in conversation, always part of a larger whole. Your health is not just your health. It is the health of your ecosystem. Your happiness is not just your happiness. It is the harmony of your microbial community. And when you suffer, when you struggle, when you feel disconnected or depressed or anxious, you are not failing as an individual. You are experiencing an ecological crisis, a breakdown in the relationships that sustain you. But ecosystems can be restored, communities can be rebuilt, balance can be regained. You are not a machine that needs to be fixed. You are a garden that needs to be tended. And the most important thing you can do, the most loving thing you can do, is to care for all the beings that make you who you are. To feed them well, to protect them, to listen to them, because they are not separate from you. They are you. You are not a single being. You are a symphony, and every note, every harmony, every rhythm is essential. So tonight, as you close your eyes and drift into sleep, remember this: you are never alone. You are always a part of something larger, something ancient, something miraculous. You are a living ecosystem, a walking universe, a federation of trillions of lives, all cooperating, all communicating, all moving together toward a shared future. And that future is unwritten. It is being composed right now by you, by them, by all of us. Rest now. Let the ecosystem do its work. Let the bacteria in your gut produce their neurotransmitters. Let the mitochondria in your cells generate their energy. Let the immune cells patrol your borders. Let the trillions of organisms that make up your being do what they do best: keep you alive. You do not have to control everything. You do not have to manage every system. You are not alone in this body. You are a parliament, and the parliament is in session. Sleep well. The Federation is watching over you.